Okay, welcome. This is section 2.5. This is our last section of this unit for chemistry. We're going to talk a little bit about chemical reactions and enzymes, and one of your worksheets is actually going through and doing some balancing equations, so hopefully you took a look at that. But let's just focus on enzymes, catalysts, and that's the primary focus of this lecture. So how do organisms carry out all those chemical reactions? They use something called enzymes. And so I've got some different examples here, and you should notice that these are macromolecules. We got protein, we got a starch, and we have fat. And the building blocks, amino acids, glucose, and then fatty acids and glycerol. So in this case, and a good indicator, like I said, glucose, OSE ending, that's an indicator that's a sugar. Well, anytime you see an ending of ASE, that indicates that that is an enzyme. So that kind of helps on pulling or distinguishing between different terms. So in this case, I've got a protein, and here's my protein chain. It's actually now just a polypeptide, but once it curls up and twists on itself, then it's a protein. So if you have an enzyme coming in, protease is going to break the protein molecules at the peptide bonds, and when it does that, it's going to break it into individual amino acids. I can look at a starch. So here's a chain that's a starch, has several glucose. So I can take the enzyme carbohydrase, break it, and have my individual glucose molecules. Then I can take a fat molecule, lipase, and break that down, and I would get a fatty acid and a glycerol. So those are examples of enzymes from macromolecule broken down into their building blocks. So an enzyme is really carried on in every cell of every plant, every human body, and it does best in an optimum temperature and even an optimum pH, which means is that, for example, when it's hot outside, you just don't do your best, but if it's around 65, 70 degrees, aren't you more efficient? So there's always, for each situation, each organism, or even an enzyme, there is an optimum temperature, and we'll look at also an optimum pH. So this is usually the structure of what a enzyme looks like, and this is kind of a simple drawing. So every living creature produces enzymes, and it will either synthesize, which means to make a molecule, or it will break a molecule. Now, I'm not going to ask you questions on how many different enzymes are in each human cell, but just to show you that each enzyme, as many as there are, they have a particular function, and they do that function over and over again. And they're very efficient because that's the only job they do. So one enzyme can process 5 million molecules in a single minute. So it's running all the time 24/7 and doing the same job over and over. So you may even see some enzymes that occur naturally. And some enzymes need something called coenzymes. So those enzymes, which would be like iron, too, those are found in vitamins. So they're needed, the coenzymes, to make any food you digest, and that's why we take vitamins. So it's this coenzyme, it kind of helps the other enzymes out. So that's one reason you take your vitamins. And we'll see at the end slide that we really use this every single day without us even realizing it. So one example is just detergent, washing our hands, shampooing, washing our pets, dishes, etc. Detergent has enzymes to help break soil particles down. Okay, so here is an example of digestion, and I was talking about optimum pH and optimum temperature. So in this case, here's my stomach, and it uses the enzyme pepsin. So this is just an example of pepsin. And it is ideal, it works the best at a pH of 2. So if you remember back to the pH chart when we were doing pH in our labs, we saw that the stomach acid has a pH of 2, or about 2. So that's where it's ideal. Okay, so if we produce too much stomach acid, okay, we usually get heartburn or indigestion. So it kind of swings and becomes more acidic. Another pH in our intestines, trypsin, it's ideal, a little more basic, and it functions the best at an 8. It'll still function between 6 and 10, but it just works more efficiently at the optimum pH. Okay, now this may seem a little confusing, but if you want to stop this and kind of look at this graph down here, it may help. So I've got my free energy, okay, on my y-axis, and it may be cut off. I've got my course of my reaction, okay. So what a catalyst does is it's going to speed up the rate of a chemical reaction. And it does that by lowering the activation energy. It doesn't change the reaction. It doesn't make new products. That's very important. So let's look at this. So here I've got my reactants. I've got my kind of red pink line and my blue line. They both start off with the same reactants, excuse me. The pink line, okay, is still going to end up and make the same product, and so is the blue line by the time it's done. Now the blue is in particular reaction, excuse me, a reaction that has not been catalyzed. There's no catalyst involved. And so it's still going to get the job done. It just takes a little bit longer. So that's what that EA means, activation energy. So it's still going to need something to get it started. So it's like when you take a match and you light the match and then you hold it to a piece of wood or paper. That's the activation energy before it starts burning. So it would be like the paper or the reactants lighting the match, and then by the time I'm done and it's burnt, I've got ashes. Those are my products. So that's just one example. Now if I look at the pink line, okay, this has been catalyzed, and you can see it's much faster. So it doesn't take as much activation energy. So it lowers the activation energy. That's what it means by lowering. Okay, here's two side by side instead of on the same chart. Same thing. So here's my pink. I've got the reactant. On the right side, I've got the reactant, and this one has an enzyme attached to it. You can see the activation energy is much lower than if it didn't have the enzyme. Now you're saying, well, wait a second, you just said a catalyst. So enzymes and catalysts, they work together. There are some similarities and some differences. I'm going to get to those in just a second. So an enzyme also lowers the activation energy. And so it's going to transform the reactant into a product, just like a catalyst. And so when you put the two together, enzyme, catalyzed reaction, okay, so that kind of, it all works together. So what it does, it just has a more efficient activation energy so that the reaction can occur more quickly and more efficiently. So here's the two tables. This is more for you. I'm not going to ask you detailed questions off of these two charts, but I think it's to help putting it side by side on how are they similar and how are they different. So how are they similar? Enzymes and catalysts. They both accelerate the rate of reaction. They both decrease the activation energy to make it run more efficiently. You can use it over and over and over again, so it has one job, both of them. And it doesn't alter the end products. You're still going to end up with the same thing if there's no enzyme or catalyst. But you want to have enzymes and catalysts. Okay, so it'd be like in detergent. Okay, if you wash your clothes, they're going to get clean if you just use water. But if you use detergent, which has enzymes in it, they'll get cleaner. Okay, you can still get the dirt out, but just not as well. So think of it that way. Now some differences. An enzyme is a pretty complex protein, so it is a protein. Catalysts are simple inorganic molecules, so they're not as complex, they're not as developed as a protein. Okay. The other thing, you can kind of bypass these two. Enzymes will catalyze specific types of reactions, whereas catalyst has a very wide range, so it can cover more things. So catalysts can do a lot more different things. Enzymes are very specific. Enzymes are greatly affected by temperature and pH. Catalysts, not so much. Okay, so let's look at a substrate. And don't think of this as a very complicated word. It's really just the reactant, and it's the site where the enzyme will bind or attach to, and they just term that substrate. So don't be confused by that. So if I come down here, this gray kind of irregular shape kind of looks like a Pac-Man on its side a little bit. So this is the substrate is this green part, two pieces. The part where it's going to fit together is the active site, and this gray is the enzyme. When the two come together and they lock together, it's now called an enzyme substrate complex. So it's going to go through a chemical reaction And what it's doing is it's going to break these two substrates apart into two products. Okay, so this would be taking like a molecule. It comes in, it goes through the chemical reaction, and now I end up with two products. Okay, so let's see, what would be a good example? If I take H2O, that's my molecule, and then I break that, and I'll end up with hydrogen and oxygen. That would be my two products. Okay, so a substrate can be transformed into one or more products, so as we demonstrated down here, and when it's released, that active site is now available to accept another molecule. So that's why it can do it so quickly, efficiently. That's all it does. Okay, in this diagram, it kind of ties in the enzymes, the substrates, and then back to our disaccharides and monosaccharides. That's why I really like this diagram. So here is my enzyme. Here is the opening for my active site. So this enzyme is called sucrase, ASE enzyme. Here I've got my substrate, okay, and this one is sucrose, OSE is sugar. When they come together, so they're going to come down and join together, that is called the active site. The sucrose will fit into that enzyme. When they come together, it's now called an enzyme substrate complex. Okay, so just putting the two together and calling it a complex. So when that happens, we have some water coming in, and then it's going to convert and it's going to break that sucrose into two different products. It's going to release the products, and now I've got fructose and glucose, and now it's available and it starts the whole process over. So remember the little mnemonic devices that I showed you? So fructose plus glucose equals sucrose. Okay? And then like I mentioned before, enzymes are important throughout our lives, so you can pause and look at this. I'm not going to ask any questions. So it could be the treatment of our wastewater, some pharmaceuticals, food, drink. I already mentioned the detergents, our cosmetics, and just taking a shower every day. There are enzymes in the soaps and shampoos that we use. The paper that you might be writing on right now. Even they put it in the like the dog food or cat food for your animal to help improve digestion. So just a lot of different things that enzymes are really important, and catalysts go along with that. So this is the last lecture for this particular unit. So again, I went through that pretty quick, so you can always pause, rewind, etc. So if you have any questions, just see me in class.